Welcome and thank you to our panelists. We have wonderful collections in this country, living collections, which you only have to think about all our botanic gardens. We have preserved collections in herbaria, the one at Kew with its spiral staircases, and of course the Linnean Society itself, which we'll hear more about. And increasingly we have digitized collections, and all sorts of things get digitized about those plants and seeds and so on. We're going to hear 4 stories today from 4 different amazing collections, and actually I think represented by rather amazing people. Sadly, the RHS couldn't join us today, but we'll be hearing from the other 4. And after we've heard from them, probably about 8 or 10 minutes each, we'll have a little discussion and then plenty of time, I hope, for questions. Please spare a thought for the moderator. It's not easy on Zoom, so my internet connection is a bit unstable. Padma will tell me if it is. And also there's a delay on the line, as there always is with Zoom. So if I cut across anyone, then it's not out of rudeness, but just out of the delay in the system. So let's get on. And the first presentation we're going to hear is from Annika Prins at Rothamsted Research. Annika, if you'd like to share your screen, and then the virtual floor is yours.
Thank you very much.
That's great. We can see that.
Okay, perfect. Right. Rothamsted has 300,000 samples of grain, straw, herbage, and soil in its collection, which goes back as far as 1843. These samples come from several experiments that were started nearly 180 years ago by John Bennett Laws and Joseph Henry Gilbert. Of the original 9 experiments, 7 continue to this day. The main objective of the original experiments was to measure the effects of inorganic fertilizers on crop yield. Each year, crop yield was measured after harvest and samples kept for chemical analysis. The different long-term experiments at Rothamsted provide a valuable resource as we can reanalyze old samples using modern techniques. In this way, old samples can tell brand new stories. This is an aerial photo of the Broadbork field, site of the oldest continuous agronomic experiment in the world. Wheat has been grown on this field every year since 1843, with different plots receiving different types and amounts of fertilizer and other treatments. Every year, samples of grain, straw, soil, and fertilizer are taken and stored in the Rothamsted archive. This is what the field looks like these days, but what did it look like back at the start of the experiment? In the early days, wheat was harvested with scythes, bound into sheaves, and threshed by hand. In contrast, we now use a small plot combine harvester for this task. It raises the question, how has wheat changed to allow this technological advance? Look at this 19th century wheat on the left compared to a modern variety on the right. One thing becomes immediately obvious. The older variety towers over its modern cousin. It stands at well over 1 meter high, while the modern variety just about reaches half that height. The development of these modern dwarf varieties revolutionized wheat production and greatly increased yields. Plants spent less energy generating tall stalks and put more energy into producing grains, amongst other things. However, there is a hidden story here that you can uncover if you know where and how to look. Even though the wheat ears look very similar between these 2 varieties, chemical analysis of wheat grains from our archive revealed big differences in the nutritional content. Now, don't worry about the detail in these graphs. The message here is that over time, from left to right on each graph, wheat varieties have not only become shorter, but have also lost protein and micronutrients like zinc and iron. Soil samples from the archive have shown that the concentration of these minerals have actually remained stable or indeed increased over time. So it's not environmental change that has caused this dilution of nutrients in the grain, but changes in the plant caused by selection. Because we selected for yield, these hidden changes occurred without us knowing about it. Now, these results have prompted research looking for varieties that are naturally higher in zinc and iron so that they can be used in commercial wheat production. This tells us how plants have changed on the inside, but what can the archive tell us about how the agricultural environment has changed over time? Look at this barley sample from 1892. The plant that this was harvested from was attacked by leaf blotch fungus, which causes one of the most destructive diseases in barley. In the mid-1970s, azole fungicides as a class of fungicide were introduced into the UK to control pathogens like leaf blotch fungus. DNA was extracted from fungus taken off this old sample and other samples from the archive to see if the fungus had resistance genes to the fungicide. Fungus from older samples did not have the resistance gene. However, samples from 1985 onwards showed that resistance genes rapidly increased. This coincided with the introduction of azole fungicides in crop production here at Rothamsted. These days, the majority of the leaf blotch fungus population at Rothamsted possesses the azole resistance gene. This has shown the need for a wide range of agronomic management tools instead of a single solution, so that the evolution of pathogen resistance is slowed down. The samples in this collection can also tell us a lot about what the world was like a long time ago and how it has changed. Once again, don't worry too much about the details in these graphs. The takeaway message from them is that man-made pollution can and have been tracked using samples from our collection. From 1843 to 1970, there was a steady increase in sulfur dioxide emissions in the UK, largely due to industrialization. And you can see that in the graph on the left, it's a solid line that goes up and then down again. Wheat samples from 1843 to 2000 were analyzed for sulfur content as well as stable sulfur isotope ratio. Now, the isotope ratio is a fingerprint for the source of that sulfur. The samples from our archive showed that the isotope ratio, and hence the source of sulfur used by plants, shifted from soil indigenous sulfurous compounds to that deposited through emissions from industry. Since emissions have dropped after 1970, the isotope ratio has also changed back to what it was. It increased again. Now, interestingly, the source of the sulfur in our samples at the peak of emissions in 1970 indicated that it came from coal stocks from collieries in southeast England and southern Wales, which is the closest collieries to Rothamsted. Forage samples from our archive have also been used to identify the presence and origin of radioactive fallout, which has traveled from faraway places like the Nevada desert, where 84 atmospheric nuclear tests were carried out between 1951 and 1962. Plutonium isotope ratios identified the source of the fallout, amongst other places, as nuclear tests by the US and UK on Bikini, Enewetak, and Christmas Island, as well as Soviet tests performed in the Arctic. Fallout from the accident at Chernobyl could also be detected in Rothamsted herbage samples based on cesium activity. And you can see that in a really small bump in the graph on the middle around 1986. When I look at our collection, I often wonder what other stories there are to tell. If you would like to delve into the data yourself, please feel free to take a look. all of the yield data and everything else is available through that QR code. It takes you to the website that's listed just underneath the QR code. And it's all freely available for anybody to look at. And that's, I'm done. Shall I stop sharing?
Yeah, sure.
Okay.
Thank you so much, Tanika. Can I just ask you, just before we from Mark Nesbitt, I wanted to ask you about sulfur dioxide. My dad told me that you know, there used to be enough sulfur dioxide in the air to actually kind of act as a fungicide and would keep mildew down. Is that rubbish, or is that has got some basis in truth?
I am not a plant pathologist, so that's a very interesting question.
Okay,
but I mean, I would assume the fact that we still have to use fungicides means that doesn't really work. It doesn't work that way.
It was limited, just briefly, I'll explain on this. It was limited to actually the impact of black spot on roses. So there was a period in parts of southeast England where sulfur was very, very high, and horticultural varieties, this is where we need the RHS, horticultural varieties of rose were being hit heavily by black spot, and the high sulfur levels did actually cause a suppression, apparently, in roses. And in some areas where sulfur is now dropping, black spot is more abundant, apparently.
Great, thank you. I was mistaken about mildew, but thank you for that. Annika, we'll leave it there, and I'll come back to you afterwards. And I know we want to explore what's going on at Rothamsted now, so we'll come back to that a bit later. Okay. And thank you, Mark, for that. That was very helpful. And now we're going over to Mark Nesbitt, somewhere in the bowels of a building, by the look of it.
Hello. So I'm Mark Nesbitt. I'm curator of the Economic Botany collection and head of interdisciplinary research here at Kew Gardens. I'm working tonight with Erin Messenger, who's just behind the camera, who is the collections manager. So we're standing in a tennis court-sized room with around about 100,000 samples of useful plants in. And economic botany in 19th century terms simply means useful, not necessarily to do with money. And that's everything from raw materials, such as timber or rubber, through to every kind of object made from plants, representing every aspect of daily life from ancient Egypt up until the current day. The collection was on display at Kew for maybe 150 years or so. It's now sitting in this large research store, and we use it for lots of purposes, for teaching, we lend to exhibitions. But it's primarily a research collection, and we're researching a whole range of topics, the histories of Kew and Britain, particularly in the last 200 years or so, reconnecting with source communities. Where did these objects come from? And I've chosen one object that I think really brings together a lot of those themes. So Erin, if you give me a hand, I'll get it out. Let's swing the lid back. Let's see. And we'll pull back the Tyvek covers. This is a spun polyester. It's really inert, really smooth. So it's a very good kind of bed for historical objects. So they're very fragile. Let's slowly. Great, thank you very much. So what we're looking at here is a taputa. So this is a poncho-like garment from Tahiti, given to Kew in 1874 by Prince Alfred, one of Queen Victoria's younger sons. It says on the label that it's made from the paper mulberry, and that these decorative elements that you see scattered across the surface are made from sugarcane leaf. And this is pretty much all that we knew about this item of clothing until two recent research projects. One, a brilliant master's degree project by Brittany Curtis, studying museum studies at University College London. We looked into the origin of the object. How did it come to Kew? What was the involvement of Prince Alfred? Prince Alfred was sent on a world tour in the 1860s as a captain in the Royal Navy. As ever, finding work for younger sons of the monarchy was always a problem, and this was seen as a diplomatic initiative and a way of keeping Prince Alfred busy. And in that tour in June 1869, he stopped at Tahiti, and this was a long anticipated visit. Tahiti, it's a kingdom, it had a royal family. Queen Komora IV, the last queen of Tahiti, was its main host rather than the French colonial authorities. He spent a lot of time with Tahitian chiefs, with royal relatives, and with the queen herself. And we know that during the 2 weeks or so that he stayed in Tahiti, from newspaper accounts, there were 2 occasions of gift giving. And gift giving is laden with symbolic importance in Polynesia. It's how you build relationships between one royal family and another royal family, for example, and particularly any relationship that involves clothing, such as a taputa, this poncho-like garment that you wear over your head. Anything that involves clothing is highly personal, brings you highly strong relationships. And we know that Prince Alfred himself is wearing one of these taputas at one of the ceremonial dinners. So this was a gift from probably the royal family to Prince Alfred, brought back to London, displayed at the South Kensington Museum, together with vast quantities of gifts acquired during this voyage, of which the Pacific component, because so much it was made from plants, was given to Kew, and then in turn redistributed in large part from Kew to many other museums all over the United Kingdom. So we now understand a lot more both about the cultural context, the gift-giving context, that's really crucial for this piece, but also its journey to Kew. A second research project that we've just wrapped up, working with the Centre for Textile Conservation in Glasgow University, the Smithsonian, and countless makers and museums in Polynesia itself, has been looking at the broader bark cloth collection here, or tapa cloth as it's often called in the Pacific. That enables us to bring together a team, not only very diverse in space and crucially really strong indigenous perspectives, but also lots of different technical abilities. So one reason I can show you this object is a beautiful packing by the research conservator of the project and the students that she taught who got experience in working with this material by preparing it. So it's really beautifully stored here, wrapped around the bolster, stabilized on the back. That means that for the first time in decades, it's possible to show this to both research visitors, makers, and indeed to yourselves. We were also able to bring a technical analyst who used lots of different techniques, for example, to identify the beautiful golden colouring on this, probably almost certainly comes from turmeric dye. We've been able to confirm, I think, but certainly the fringe here, you can't really see, but imagine paper-like, tissue paper-like fringe on this taputa is made from the cuticles, the upper part of sugarcane leaves. And this would have been worn for dancing. It would have rustled and shaked as you dance. And the decorations here are probably from banana leaves, sugarcane leaves. In fact, we know how almost to the day when and where it was collected, June 1869 in Tahiti. It's really important to understand this, because what you're seeing is a fabric that we now know not to be made from paper mulberry, but almost certainly from breadfruit, the really ancient bark cloth of the Pacific, replaced in the 18th and 19th centuries by paper mulberry, the main plant that's used today. And we know it comes from the time 1869 when tapa production was reducing in Polynesia, when woven fabrics in Manchester and elsewhere were arriving into the region, replacing the really labor-intensive process of beating the inner bark of trees in the mulberry family to make bark cloth. So this is a crucially important document of the almost the end of tapa production before its revival in the 1950s and 1960s in the region, particularly in the islands that have stopped making it as a really important element of local cultural practice. And so what you can see here, reflecting that late date, is a millennia-old tradition of making bark cloth, overlaid with European style decoration in the form of these rosettes. This is very much a hybrid or fusion object that represents the encounter of indigenous cultures in Polynesia and the arrival of Western travelers, explorer, colonization and trade. So we have 100,000 objects in this collection, and it's my ambition, not in my lifetime, but to make sure this process continues of exploring this collection and thinking about its significance, not only in a Kew context, but most of all in the context of its importance to source communities. Thank you very much, and back to you, John.
Thank you. Just before you go,
Yep.
He's already gone. Oh, there you are. Mark, can I just ask two things? One, Erin, I don't know whether you could just give us a close-up or just have a go at giving us a close-up.
Yeah, might not be a very smooth transition though.
Yep,
no, don't worry. Yeah, I just would love to see. And while she's doing that, Mark,
yeah.
I don't know whether that's.
And we can see the neck hole just up here.
Fantastic.
Yep.
Yeah. Oh, it's beautiful.
And then the
Thank
fringe
you.
down here with
And Mark,
cuticles.
and just tell us how that cloth is made, I guess. What's the technique they use?
So trees such as breadfruit or paper mulberry, they're coppiced. So you harvest them at about 2 years old when the branches are still very young, about a finger's width or a thumb's width. And the inner bark, which is a phloem tube carrying the sugars from the leaves down to the roots, is still very soft. And it's then relatively easy to separate out the inner bark from the corky outer bark. You have end up these long strips, perhaps 6 or 8 feet long, of the inner bark, and you beat those. And as you beat them with your wooden beaters on an anvil, they spread to perhaps 6 or 8 inches wide, a handspan's width. And when you beat those strips together, perhaps using a bit of adhesive, perhaps from a fruit or a sap, and you can make sheets of any size. So these are used for all forms of clothing, for room dividers, for all kinds of textile purposes.
Fantastic. Thank you so much, Mark. Now I think you're going to scamper back to your other microphone, your other camera, and we'll come back to you a bit later. But thank you, and thank you, Erin. That's great camera work. Thank you. Fantastic. Next, we're going to go to Zoe from Botanic Gardens, Edinburgh, and I think you're going to tell us about that. So do share your screen.
Right, there you go. That should be working. Can you see everything now?
Yeah, I'm good.
Thank you. So, good evening. I'm going to try and follow that amazing cloth. So this evening, I'm going to talk about a plant specimen. So unlike the previous talks where we're talking about a lot of historical collections, I'm going to talk about a plant specimen that I collected just before COVID-19 hit. So in February 2020, I was surveying the dry forests of the isolated and stunning Marañón Valley in the Andes of northern Peru. On the left here, you can see where Peru is located in South America, and in the larger elevation map, you can see a steep valley in the north highlighted. This is the Marañón Valley. For this project, I was working with Paul Gonzalez, an up-and-coming Peruvian researcher from the University of San Marcos in Lima, and someone who can manage to look very suave while riding a mule. For our project, we wanted to record and identify every plant species found in the valley to produce the first comprehensive species list for this forest. The valley has been understudied, but what we knew from before the project suggested that the forest is full of species which occur nowhere else on Earth. The Northern Andes are considered to be one of the world's most diverse biodiversity hotspots, and isolated valleys like this one may explain why. We call the habitat in this valley tropical dry forest. This means it rains for only 2 to 4 months a year and is hot and completely dry for the rest. Yet it contains fully grown trees and shrubs, but also large spectacular cacti and succulents. Usually this forest has a complete canopy, but here in this photograph, the mountainsides are so steep, it looks more like a shrubland. In early February 2020, as COVID started to sweep the world, we were collecting plants at this very spot, a glamorous roadside next to an electricity pylon. It was here that we collected this plant. Now, luckily, Paul Gonzalez is also an expert in this genus and was able to identify this plant in the field as Solanum arcanum. It turns out that this species is a close relative of the cultivated tomato and was described for Science in 2005 by Iris Peralta, an Argentinian expert. It is one of dozens of species which are close wild relatives of the tomato found in the Andes, the region where the tomato was originally domesticated. On the left here, we see the specimen that we collected, and on the right, we can see some close-up images of the living plant. You can see it looks very much like a domesticated tomato, albeit one which looks a bit leggy with slightly smaller fruit than usual, but recognizably a tomato or something very similar. If you are not familiar with the genus Solanum, it includes the tomato, the potato, the eggplant or aubergine, and many other lesser-known crops from around the world. You can see from these habitat shots from other specimens that this species is restricted to extremely arid places with lots of bare soil and rock across the coastal regions and dry valleys of Peru. This might seem odd to us given the huge amount of water and extremely fertile soil required for cultivated tomatoes to grow. But this is what is so interesting. Tomato wild relatives have already contributed to our cultivated tomato. They are responsible for large increases in the size and sugar content of the tomato as we now know it. But wild relatives are also a major source for bringing in traits for resistance to external stresses, such as drought or salinity, as well as pest and pathogen resistance traits. So judging by its habitat, Solanum arcanum clearly has a tolerance to drought that could be very useful in the domesticated tomato. But crossing is a lottery. Initial crossing with Solanum arcanum, in fact, gave domesticated tomatoes cold resistance and resistance to bacterial and fungal nematode diseases. Not at all what we expected. So while on that day in February 2020, Paul and I were interested in the unexplored flora of the amazing Marañón Valley, but amongst the plants that we collected were specimens like this Solanum arcanum that researchers like Iris and Paul continue to study for their importance as wild relatives to globally important crops. This research will allow us to further use these wild relatives in tomato breeding in the future. By incorporating traits from these tough wild plants, we give ourselves a chance of meeting the increasing demands for food under a rapidly changing climate. This is also why fundamental research of recording the plants that grow in extreme places like the Marañón is so necessary. Our project is still discovering species new to science in places like this, but also new collections of known species like Solanum arcanum in challenging environments are important as traits vary immensely between plants of the same species in different places. And none of these discoveries are possible without these collections. Sorry, I might have rattled through that slightly.
No, that was so interesting. Thank you. You mentioned that the sort of breeding doesn't always give you what you think you're going to get. How are we going to get drought resistance? What's the sort of procedure to get that quality? My tomatoes are completely thirsty.
Yeah, well, I guess the short answer is complicated, as shown by the fact that the initial crossing failed. I think part of the problem, so basically we're now at this amazing point where we have all these specimens of species like Solanum arcanum from all these different places across their range. So for the first time actually in history, we know where they live, we know what conditions they live in and what physical characteristics they have. And so it's only now, especially with collecting seed from these populations, that researchers are able to go out and basically get good quality DNA from across all these species, across all their different habitats, and then work out what genes are at play. And one of the things that seems to be coming across is that drought resistance is not just one gene, it's a whole suite of different genes. So this is why it's not coming across in a straight crossing event, because you require multiple genes. And then the second thing that appears to be happening is that, so for example, straight drought requires one set of genes, but if you have drought plus heat, it isn't just a case of your heat tolerance genes plus your drought tolerance genes. It might actually be a third set of genes entirely that you need. So it's not additive, it's very different to that. So I guess there's two possible futures. One is if we know what genes look like in a drought tolerant plant in the wild, you can maybe look in the domesticated tomato for the equivalent genes and just switch them to the right format. And probably what's more likely is that you can use these kind of marker screening techniques. So just do traditional crossing, but in your thousands of baby tomato cross F1 hybrids, you can use quick marker screening to find the offspring that have carried over the drought genes, and then back cross those into domesticated tomato to get those genes, those suites of genes in. But as I said, it's not even additive. So if you're looking for somewhere hot and dry or cold and dry, it might be a whole different set of genes again. But it's only now that really we've got the tools to kind of work out what's going on. And it's through good collections. You need to know what plant it is you're looking at, and you need seeds that to produce the really high quality DNA. Jonathan.
Thank you.
Yeah, he's back.
Oh, am I back? Oh gosh, that was a heart in the mouth moment. Thank you very much. So I could hear you, so but even if you couldn't hear me, with the Linnean, and very, very pleased to have you with us, Mark. And perhaps just a line about what you do, and then the floor's yours, and share when you want to.
Thank you very much for that introduction, Jonathan. My name is Mark Spencer, and amongst other things in my life, I am the honorary curator of the Linnean Botany collections, which I will be talking about in a moment. So I shall just set my screen up for you. And I am going to be talking about taro, but this is a tale potentially that I could be telling about one of many plants that are very important to human well-being globally. And I've just accidentally moved that forward. And I'm just going to hide this because it's annoying me. There we go. So this is a brief history of taro, told really through historic botany collections and also historic botanical literature, to tell a story about how we can understand the domestication and movement of plants around the world, and how this is not only fascinating scientifically and culturally and historically, but is also very, very important ultimately when it comes to the challenges that we face in the world today around environmental change and the impacts of pests and diseases, for example. So here I have an image from an early work in the history of botanical literature produced by a gentleman called Matthias Lobel. And this was published in 1581. And it is this image is one of many woodcut prints in this particular work, dealing with plants as known by principally the European world at this time. You'll notice that taro at the top is actually given the name Arum aegypticum, meaning that it's believed to be a type of arum and that it hails from Egypt. So here we have a plant which is essentially known to the European north as a plant that is related to a wild plant, an arum, lords and ladies of our landscape and environment, but is, as we believe it and understand it, to be from Egypt. But the story is more layered and more complicated than that, because taro is a plant that originates from South and East Asia. You can see on this map, which I gleaned a few hours ago from the wonderful Hughes Plants of the World online resource, a map of its wild distribution in South and East Asia. Now, taro today has been moved around the world and is found in many, many parts of the warm, temperate and tropical globe. In fact, this map is probably an underestimate of its distribution in the purple parts of the map. So why is taro important? Taro is one of the key sources in particular communities around the world for starchy root crop, and is particularly important in parts of Western Africa and also in South and East Asia and Oceania, and for example in the Maori culture of New Zealand. This is a plant that has been spread around the world for millennia. It has one of the longest known origins of domestication and use in agriculture of any plant. Now, it's, as you can imagine, with such a long history for such a species in human life, its naming history is very, very complicated and diverse. And by the naming history in this particular context, I'm talking about the vernacular names that we give to plants in our various societies. Generally around the globe, this plant is now known as taro, but it is under many, many other names, for example, dasheen and other such names. Now, I'm just going to allude to a very useful and interesting paper and a series of papers written by a lady called Ilaria Grimaldi, who's done a huge amount of work along with colleagues on the history of language and domestication and uses of agricultural plants such as this around the globe. And you can have a further look at this later on if you wish. So taro is now a globally important crop that has moved from East Asia into the various parts of the world. It was known in the Middle East and Europe from antiquity. In fact, its name Colocasia is actually in origin, in part, actually ancient Greek. So this is a plant with a deeply rich linguistic history. Now I'm going to move sideways away from talking about the deeper history of this plant and talk about the more recent recorded literature and modern scientific history of this particular plant. And I'm going to be doing it through the lens and the history of the Linnean Society of London. This is Carl Linnaeus, a very, very important man in our lives because his personal collections and materials are part of the foundational materials of the Linnean Society. And he is a man who is of immense importance to the rest of the world. He is probably the scientist that many people have not heard of, but our lives are very, very dependent upon the innovations that he created, because core amongst those was the creation of the binomial system. The fact that we are called Homo sapiens is principally driven through the work and the activities of Carl Linnaeus. So this gentleman from Sweden is an immensely important man in the history of biological science and a personal hero of mine. Within the Linnean Society's vault, we are incredibly lucky to have his collections, which were purchased by our founder, James Edward Smith, in the 1780s, and they're now in this environmentally controlled vault. The volumes you can see at the top are actually his personal publications and library and literature, and the bundles underneath are his herbarium, his plant collection. There are approximately 14,000 plant specimens in here, many of which are representatives of the some 9,000 or so plant names that he devised throughout his scientific life. So, let's move forward. I'm going to use that. Excuse me, my screen is right. So one of the things that is immensely important about Linnaeus is not only his creation of the binomial system. This plant here is Agremonia eupatoria, a member of the rose family, a widespread plant in Britain and Northern Europe, but it's also his approach and methodology to presenting information about plants. Here on the right-hand side, we have the original type specimen, as we call it. This is the physical object upon which the scientific name Agremonia eupatoria, the scientific name for this plant, is based. And here on the left-hand side is a plant specimen I collected from Charles Darwin's backyard some 15 years ago, which is prepared and mounted in very much the same way as in Linnaeus's period. So methodologically, the innovations of Linnaeus and his peers helped create the way we do modern botanical science today. And that is not just about the methodological approach, but it's also about actually how these specimens are immensely important. We've just been listening to a fantastic talk about crop wild relatives and modern exploration in southern South America, in the Andes, looking at crop wild relatives of the tomato, Solanum lycopersicum. And here we have on the right-hand side, Linnaeus's own specimen of tomato. This is the basis, the type or lectotype for that scientific name. And on the left, we have an incredibly important plant globally. This is rice, or Rhiza sativa. So these are physical objects which pin down our understanding of scientific names and are potentially also very much active scientific research objects, which I will turn to a little bit later on. Now, rather curiously, the type for Arum esculentum, which is what we call the base name or basionym for Colocasia esculenta, is not a specimen. It is this illustration produced in Sir Hans Sloane's work, Voyages to Jamaica, which was published in 2 parts in 1707. This is culturally and historically a very, very important work for the Caribbean, particularly Jamaica, because it is full of some of the earliest descriptions of the plant life and natural history of Jamaica. It is also full of some very, very challenging commentary and information on the treatment of enslaved peoples and their culture and lives and the experiences they went through. So this is an incredibly important work which speaks volumes about European understanding of plant diversity and is also, as I say, the basis of the understanding of this plant, Arum esculentum, or as we now know it, Colocasia esculenta and taro. So it is a rather unusual type specimen. Now, I mentioned earlier on that actually this is a plant as a wild plant from South and East Asia. It's a part of the world where it does actually have about another 6 or 7 crop wild relative species around their other species of Colocasia. This particular early work by Rietod van Strackenstein, Hortus indicus malabaricus, is a beautiful work, but also incredibly important because it is some of the earliest representations of South and East Asian natural history and botany and art, but it is also full, and rather unusually for the era, with primary information about the names of the communities in which these plants were growing. Up here on the right-hand side, there are names in Tamil, Malayalam, and other languages. So this is an incredibly important document about the plants, but also the interactions with those cultures.
Mark, that's 10 minutes.
Oh gosh, I'm really sorry, I didn't realize. I'll be really quick. Terribly sorry, I didn't realize.
Rather than rushing through things, why not?
You've cut out. Terribly sorry.
Take the time just to tell us a bit less of what you got.
Yeah.
But,
So European perceptions about this plant shifted towards actually looking at plants from an ornamental perspective, such as this. And then further on, as we move into the Americas, we find this plant actually becoming part of the life of the Americas through the Atlantic slave trade, as presented by this image here by Plumier. And further on, I wanted to talk momentarily about this particular specimen, which is one of the key elements within the history of taro, these are plant specimens from Linnaeus's herbarium, and these were in part collected from a slave plantation in Suriname in South America. But the reason these plants are very useful and important is because the understanding of history of domestication and movement of crops around the world helps us understand how domestication affects crop diversity. And this can also be incredibly powerful and important when dealing with challenges such as taro leaf blight, which is a very, very important and serious disease to taro around the world. And terribly sorry for overrunning. I got overexcited as usual.
Oh, you've hardly overrun. I feel terrible now. And absolutely fascinating, and how I was going to say how lovely, but I don't mean how lovely, but how interesting to finish on the leaf blight there for Plant Health Week. So thank you for that. One of the things that I'm just sort of struck by is just the sort of range of uses of the such an incredible range of both scientific and cultural stories. Very exciting. I wanted to ask you, as a group, before we go to some individual questions that people have posed, about the kind of next stages. What happens next with research in your different organisations? You know, where to go from here, and what's going on at Rothamsted with the collections? What are the sort of next questions that you want to try and answer?
Yeah, so at Rothamsted, so there are two main current things that I wanted to highlight. We've actually been contacted by some students from Lancaster University, and they're interested in tracking the presence of microplastics in our crops over time, because they do actually, they show up in our crops, and obviously we don't want to ingest these things. So that's a current usage of our collection. And then secondly, we have yield data, but we've got information about all of the inputs, all of the fertilizer. We also have a small weather station on site. So we've got precipitation, we've got sunlight hours, temperature, and all of this information can be put into like a model that can be used to predict yield, which would be really valuable to farmers. It's always about the yield. But yeah, so that's two of the ways it's being used.
Fantastic, thank you. I'll come to Mark Nesbitt next, and then Zoe after that. Mark.
So could you repeat the question?
Future uses.
Yeah. So.
Oh, sorry. Yeah. We're just talking about the sort of future uses of the various kind of collections that we've got. And you know, where do you go from here? What are the sort of questions that you want to answer, or what are the kind of the directions that you think this will go in?
Yeah, I mean, I think Aneka made a really good point about new techniques open up new uses for these collections. So, for example, we have 15,000 fruits and seeds in the economic botany collection, but all dead, but we're not frozen like in our seed bank. So you might have said even 20 years ago, but these are useless. You can't germinate them, can't do anything with them. Now, thanks to next generation sequencing, we can get fragments of DNA, even very small fragments of DNA out of the specimens. We've just published work, for example, on an ancient Egyptian date palm, looking at the evolution of date palms. So I think now I obviously have let's say the next 10 years or so in my mind, very roughly planned out. So we're doing a big project in Brazil, in the Amazon, for example, with source communities. But I think what's really exciting is that thanks to WizKids developing new machines and new ways of analyzing those results, we don't know the potential of these collections. But it's a hard thing sometimes to explain to managers, to bean counters. But we cannot quantify the value of these collections in future, except we know they will be highly valuable.
Thank you. I love the idea of being able to predict your life 8 to 10 years ahead. Zoe, where's your?
Yeah, I mean, a similar theme, I guess. I guess, as ever,
research or your organisation is looking.
Yeah, as ever, like RBGE, so the Royal Botanic Garden, Edinburgh, our primary function is just collecting, identifying and naming plants. But that leads in, it does lead into so many avenues. And what's kind of been interesting is there's obviously been this big push the last 10 years to digitize our collections. Maybe we're getting there very slowly. And it's not the digitization that's the interesting thing. It's like the avenues that it's opened up, because people who would never have accessed our collections before are starting to access them. So whether that's artists or scientists from completely different research backgrounds, I'd say one of the big things that maybe is starting to come up here is that is this, identifying, is using collections from plots in forests, so especially tropical forests where a lot of the sort of carbon is stored. It's not just about getting the right name on those species, but maybe using herbarium specimens to look at traits that those trees have. And so then we can start to predict, well, how are these trees going to really respond to the changing climate? And so if you're talking about modeling, you're talking about modeling carbon dynamics and how species individually are going to react to the Amazon forest changing. Well, we can look for clues of that both in the DNA, but also in the traits that those specimens have. And we can see already maybe how, certainly how the traits on the plants themselves have changed over time. Because remember, each of these collections is a plant from a point in time and space. So if, as Annika's work highlighted, if you take multiple points through time, you can see these traits changing in amazing ways, but also sadly to the way the climate has already changed, and hopefully will give us a clue as to what's going to happen, and we can sort of feed that into our models.
Thank you. And Mark, where's the Linnaean's research going?
Well, the Linnean Society is in a rather unusual position, and we're a small independent charity and don't as such, and learning society who don't as such do research simply because of capacity. Our drive is to enable and connect people with ideas and avenues around research. One of the key things is actually connecting with the scientific community and explaining that our historic collections are not sacred and never to be touched. They should be and are working materials to be handled and utilized for DNA extraction. So, for example, I've given in recent years permission to have DNA extracted of specimens of apple, taro, as just mentioned, and also sweet potato, to name but a few. So these are active working collections. Digitization was very, very important, but it has, and it's offered many options, but it's left some sort of rather avenues which are a little bit frustrating. We probably failed to learn in the very early years of digitization within the scientific community how collections could be immensely valuable for other disciplines, the arts and humanities. And we are now playing catch up with finding ways of getting the information in, connecting it with those communities, and critically receiving information back. The amount of times, and I can see nodding from my peers, scientists learn from people from source communities, from indigenous communities, and people who are practitioners in the arts and humanities. So we need to find better and more intelligent ways of connecting data across the disciplines.
Thank you. And of course, to sort of state the bleeding obvious, when you're digitizing.
Terribly sorry, you stopped, couldn't hear you.
There was a phase when people were just digitizing. Okay, so can you hear me now?
Yeah, I think one of the things is we initially digitized because we felt that the physical representation of the object was paramount, which is clearly very important. Particularly with early modern and historic objects, we failed to appreciate early on in the digitization revolution that the information and materials relating to that object are not often on the actual herbarium specimen, which is usually the case for a well-presented modern specimen. They're in the correspondence, the archives, and the associated literature. So we've now got a sort of unholy triumvirate of materials scattered across source materials that are incredibly challenging for historic and early modern collections.
Now Don Drake has asked the question, which is I think a very interesting one. Where are the gaps in the collections? What are the things that you sort of look back and you think, oh, if only they had collected something. I sense Mark in your Mark Nesbitt in your talk. You said, unfortunately, I seem to remember you saying there were certain elected at the time, and you've had to together in a kind of detective way. What do you wish they'd known then?
Well, I think allows me to talk about another piece of work, the work of indigenous Indians in the Northwest Amazon, really foundational for this work. I'm sure that Mark and Annika and Zoe echo this, is understanding your collections. You need to know the when and the where in order to know where the source community is, in order to really inform that engagement. And research by another master's student, bringing together all of these widely scattered sources that Mark talked about digitally into a spreadsheet has shown that Richard Spruce, the extraordinary collector who all my fellow speakers will know, who assembled both ethnobotanical collections and herbarium specimens at Kew, he connected the two. His ethnobotanical collections, poisoned arrows and so on, have numbers not with them, but in other places and journals and scattered notes, but connect them to herbarium specimens, but allow us to verify the identity of the plants used in those objects. Spruce never published that technique, and it was rediscovered by ethnobotanists in the 1980s. And if only he had set out his methodology, it would have led to the hugely improved quality in museum collections in the sort of following 150 years.
Thank you. Zoe, Annika, do you want to, that you wish were around and had been collected at the time, or sort of huge gaps? Annika?
So for me personally, I have a big interest in crop wild relatives. We don't have any crop wild relatives in our collection here, but we have access to it elsewhere. So it's not necessarily a thing that doesn't exist. It's just that it's not on site for me to use. That's all I can think of, really.
I'll tell you what that I wish someone had collected is silphium, which is that wonderful plant that the Romans used to sort of wax lyrical about and stick in all their food. The nearest relative that we think there is Acer fetida, that rather appalling stuff that actually smells quite nice once you start frying it in Indian food. But that went extinct in the silphium went extinct probably in the sort of 1st or 2nd century. And I wish someone had collected some of that, or at least documented it properly so we could work out what is. Zoe, anything from you?
Yeah, I guess, because we're still really exploring our collections, because I mean, here at Edinburgh, we have over 3 million herbarium specimens alone. There's clearly like a lot of stuff to tell. I guess sort of playing on Mark's theme, I do wish that we still had those records in his example of spruce, because sometimes you come across these collectors, you've never heard of them before, but there'll be a lengthy note on the specimen saying, this was collected the day after this volcano exploded in blah, 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 blah place in Colombia. And you're like, tell me more. So that person very kindly really gave us context. But very often the older collectors, they're not there, they're a bit vague on their dates, they're very vague on their locations. So it takes a lot of detective work now to work out where they were collecting, apart from this guy who talked about a volcano exploding the day before, which was pretty specific.
Just before I come to you, Mark Spencer, Zoe, while you're there, Kevin Doyle has an interesting question specifically on tomatoes, which is it possible that drought resistance and large fruits are actually mutually exclusive traits?
It could well be. I don't know anything about the genetics. I'm very much what plant grows in what place. But I mean, the answer is it looks so complicated. It could well be mutually exclusive. I mean, what has come across so far just from random crossing is you get a lot of that sugar and large fruit size came from plants that absolutely didn't have those qualities in the wild. But when a set of genes came over in a crossing, that's the characteristics that the domesticated tomatoes then appeared with. So we're gonna, I guess, give us another 5 years, give researchers another 5 years, and they will have a really good idea of what's going on and why we're getting these very random results from normal crossing events. But yes, it could well be that one is blocking the other. I suspect It's mainly just that drought genes are there's just so many of them. It's not a single gene. So, and then you're placing them in this different genetic environment. So, it could well be something there is blocking them and making that transition very difficult.
Thank you. And Mark, what do you wish had been collected or the collection somewhere in the world and they're just not there?
Well, the first thing is that the Linnean Society collection is rather unusual in that it's a closed collection because it's a historic collection. We no longer add to it. What I wish is that Linnaeus's herbarium, his personal herbarium, which we are the inheritors of, had not been left in the garden shed in Hammerby for a number of years and declined and decayed and got the mold. And when his son Linnaeus Filius took it over, was obliged to throw away many hundreds of specimens. So we lost a huge amount of information about Linnaeus's understanding of plants. And the thing that really surprises people is we have 14,000 Linnaean specimens. We have another 28 or so thousand incredibly important specimens that were collected under the collection of James Edward Smith, including very, very valuable historically collections from Banks and Solander, from New Zealand, Australia, etc. Many of those specimens, and this is a surprise to many, have still not been critically identified or determined, as we like to say in the world of botany, by a botanist in about the last 200 years. So I would entreat people from all around the world who have an interest and knowledge of plants from the world is look at our collections and give us your determinations.
Thank you, Mark. Now, one of the things, Mark, you just brought up was, you wished that hadn't been left to get mouldy and whatnot. I think it'd be interesting to talk a little bit about how these things have actually survived. A lot of the samples are very old. Annika, you showed a photograph. In fact, I think it's there in the background, of, just behind you, of bottles which appear to be just on a shelf. Maybe your collection, from the 1880s or whatever. I only have to store stuff in the cupboard for a bit, and it seems to go mouldy. How's your samples been surviving?
So they are just kept in a building. It's at the moment not even controlled for humidity. There is no heating or cooling in this building, but the samples themselves are dried out. So you can't sow these grains and get a new wheat plant from it. So they're dried in the oven. Everything's dried. The soil is air dried. And then, they get a cork at the top. These days, samples are put in metal tins like this. I don't know, it works.
Okay, thank you. And Mark Nesbitt, I remember you showing me around when I was with Q, around the economic botany collection and being absolutely frozen.
Yeah, so
while we looked around this fantastic museum.
You're cutting out.
Yes, I'm sorry. So maybe you could say something about the kind of techniques that are used to store various samples at Kew.
Yeah, so we're very lucky, really, compared to people that have got animal collections that have proteins in and are quite unstable. The main ingredient in economic botany collection is cellulose, so that's what it's the foundation of clothing or paper and books, but it can be 600 years old and in perfectly good condition. Our oldest object is an ancient Egyptian basket, it's around about 6,000 years old. So the great enemies of cellulose are moisture and insects. So we're careful about leaks and we are careful about insects. I think I was absolutely sure that everyone else in this call will agree that the only answer to pest damage is constant vigilance and use. And actually the collections I've seen destroyed by pests and other institutions outside the UK are collections that were mothballed, but without mothballs. So they were simply put into storage to save money or whatever. The door was opened 10 years later and lots of beetles flew out and the collections had gone. So we, of course, have we have little sticky traps everywhere. but we also really rely on users being in the collection with sharp eyes open, reporting to us the moment they see probably not an insect. You're more likely to see frass, but a little bit of dust, but it comes out of the holes. And of course, our tool now for controlling insects is not pesticides, it's not fumigation, it's deep freezing, which is such an important message to share to people at home. But if you do get moths in your jumper or bugs in your flower, pop it in your deep freezer, probably for one you've got at home, 2 weeks is about the right length of time, and it will sort out your problem for you. But we have much more powerful deep freezers, so we can do this much quicker.
I had no idea that you needed to put the just overnight would be enough, but probably insulates too much, doesn't it? Thank you. Now, there are a few questions from people in the audience who they're for individuals. So I'm going to come to you, Annika, first. And there's a question about from Rachel S. Curious if the nutrient declines have also been examined in the context of rising CO2 as well as varietal nutrient uptake. That might be something off the top of your head, but is that something that you've researched?
Well, so,
as you know,
wheat has a type of photosynthesis that really likes more CO2. The more CO2 you give it, the more carbohydrate it will make. Other crops like maize doesn't really care. You can give it as much CO2 as you like and it will not produce more carbohydrate. So in that sense, you know, more CO2 is good for wheat. If it didn't come along with climate change and higher temperatures and everything else, it would be fine, but it isn't. I'm not really sure how that links in with the nutrient decline. The total amount of nutrient in the grain is actually the same, but there's more starch in the grain. So it just gets diluted. We eat more starch. And so we're looking at other varieties that have, I guess, higher natural content of iron and zinc. They do exist, and I've seen a few papers about them, but they come at a yield loss. And so selling that to a farmer is always a hard sell. So you try and isolate that high nutrient, micronutrient trait and cross a high yielding variety with that. But it takes quite a long time to get these things together if you're not allowed to do genetic engineering.
We're nearing the end of our time, and I'd like to come to each of you in a second, just to ask you kind of if you were showing your you know, your most wonderful thing in your collection that you would want to show them. And it may be something that you've already shown, Nesbit, which is about the use of turmeric, from Caroline Hyde Brown, who asks, I think you mentioned turmeric in the context of the breadfruit and bark cloth. Were they using some traditional dye techniques, and did they dry their?
Terribly sorry, Jonathan.
Or,
we can barely hear you. We're getting about one word in every 30 at the moment.
But I'll give a quick answer to the question around traditional dyes. Caroline and others, we produced a wonderful open access book. If you visit the Kew Institutional Repository or my own personal website, you can download the whole PDF. It's open access, a really important part of the knowledge sharing, but I can see it's coming up in the chat bar. There's also a great website. If you search Tapper Glasgow, in amongst many Tapper spas, you will find Glasgow University's website presenting the results of our bark project with loads of information about our results. So open access and sharing our work, as Annika stressed in her talk, is a really important component these days.
And if you can say in one breath, please, in one breath, your sort of favorite thing in your collection, in one sentence or one breath. Mark.
Which Mark you talking to?
You go
Mark
ahead.
Spencer.
Sorry, I
Mark
made
Spencer.
Going on the climate theme that Annika was addressing, collections are very valuable tools for understanding the impacts of climate change. And as a consequence, every single botanical or biological object in a museum is of immense value to all of us. This is the driving thing we need to be considering globally at the moment. And the UK's natural history and botanical collections are one of the greatest tools globally that we're not using enough to help understand the challenges that we face.
Thank you, Mark. Mark Nesbitt.
Well, our Japanese paper collection is perhaps the finest in the world. It's extraordinarily beautiful, has that wonderful combination of craft and art, and is a continuing tradition. And we are in contact with the descendants of the makers of a large part of our 1870 paper collection. So that direct continuity to the present, what that enables you to do is really exciting.
That's fantastic. Thank you, Zoe, and then finally Annika. Zoe.
Yeah, I'm going to slightly cheat and say all of it, because it's, for me, what's really amazing is when you start to open those cupboards and look at these specimens, you really start to think, hang on a minute, wow, diversity, like that is a real thing. And I think, you know, as an undergraduate, you learn about diversity, but you never fully appreciate it. But something like a herbarium, especially these international collections where you get specimens from all around the world, you start to think, wow, like there are a lot of plants out there, and they're, you know, yeah, diversity.
Thank you. In a way that you and Mark have given two sides of the same, Mark Spencer have given two sides of the same coin, that it's actually the power is in the collection rather than an individual thing. So perhaps I've done a bad thing in asking you for an individual thing there.
I think Annika, you should just...
And finally, Annika.
So I think my favorite thing about our collection is actually that it shows you crops over time, but it also shows you the connection with the people who collected all of the samples. You get beautiful handwriting from a really long time ago, the labels. Also, something like during the Second World War, containers were running low. There wasn't enough glass. So people actually donated tins from the Ministry of Food, so the national dried milk given out as rations. When it was empty, the containers were donated to Rothamsted to put grain inside it. So that's...
Thank you. Look, we're time. Sorry if I'm across anyone my internet connection. Thank you, Mark Spencer. Thank you, Mark Nesbitt, and thank you, Annika, for coming to talk to us. I mean, really fascinating, and thank you to all the people who've sent in questions and the people in the audience. I hope you've enjoyed this. I'm going to hand back to Padma.